Have you ever wondered if the color of your eyes, or even the exact height you might eventually reach, could be predicted before you're even born? It honestly sounds like straight up science fiction, right? But it's actually just the fascinating reality of biology. Welcome to today's Explainer on Genetics, where we're going to show you exactly how simple tools, like Punnett squares, hold the ultimate key to understanding your genetic destiny. Let's get into it.
Our focus today is unlocking Mendelian genetics through Punnett squares. Now, mastering this mechanism is absolutely crucial, especially if you're gearing up for those O-level biology exams, because this exact concept carries some serious weight when it comes to marks. But honestly, it goes way beyond the classroom. Getting a firm grip on these fundamental principles of heredity is a real game changer for understanding real-world applications in medicine, agriculture, and a whole lot more.
Okay, here's our game plan for today. 1. The blueprint of heredity. 2. Mastering genetic vocabulary. 3. First-generation Punnett crosses. 4. Second-generation phenotypic ratios. 5. Avoiding common exam mistakes. And 6. Our final recap and tips. Section 1. The blueprint of heredity. Let's start with the absolute basics here. Heredity is simply the biological process where living organisms pass down their characteristics to their offspring. It's the literal answer to why kids often look so much like their parents. The scientific study of this specific process, well, that's what we call genetics. Through genetics, we explore exactly how traits are inherited, the precise mechanisms running behind the scenes, and the variations that naturally pop up within species. And that leads us perfectly into Mendelian inheritance. Gregor Mendel, with his famously obsessive experiments on pea plants, laid the complete groundwork for everything we currently know about monohybrid inheritance. Basically, this is the specific study of how a single, isolated characteristic is passed down from one generation to the very next. Section 2. Mastering genetic vocabulary. Okay, to successfully map out these biological equations, we first need to explicitly define the exact components we're working with.
Let's kick things off with progeny. This is just the technical term for the offspring produced from a genetic cross. Whenever we map out one of our genetic equations today, the progeny is the biological end result we're trying to predict.
Next up, we have the purebred. A purebred individual is homozygous for a particular trait. Now, what does that actually mean? It means they possess two absolutely identical alleles. Take a look at the variables here. It's either two dominant capital T's or two recessive lowercase t's. They perfectly match. In direct contrast to that, we have the hybrid. Hybrids are heterozygous. This means they possess two entirely different alleles for a specific trait. Notice how we have a dominant capital T sitting right next to a recessive lowercase t. They're mixed. Section 3. First-generation Punnett crosses. All right, let's shift from concept over to application. We're going to set up our very first monohybrid cross between a homozygous dominant tall plant and a homozygous recessive dwarf plant. To pull this off without making a single mistake, we need to treat the process like a step-by-step machine. Step 1. We identify the parental genotypes. Here, those are capital TT and lowercase tt. Step 2. We determine the parental gametes. Watch how they segregate into a single capital T and a single lowercase t. And finally, step 3, we actually draw out the Punnett square grid so we can map their exact intersection. Now trace the movement here. As the capital T gametes from one parent drop in to combine with the lowercase t gametes from the other, they fill out our grid. So the crucial point to see is that in every single intersecting box, the combined result is exactly the same. Capital T, lowercase t. This tells us with absolute mathematical certainty that all F1 generation progeny have that hybrid genotype TT. And because that capital T is dominant, every single one of them is phenotypically tall. Section 4. Second-generation phenotypic ratios. So what actually happens if we push this experiment just a little bit further? Let's take two of those heterozygous F1 generation plants we literally just created, meaning we're crossing a TT with another TT, to find our F2 progeny. Let's watch how this builds out our second-generation grid. A capital T and a capital T meet in the top left to create a purebred TT. Then, a capital T and a lowercase t intersect in two different cells, creating two of those TT hybrids. And finally, tucked away in the bottom right, two lowercase t's combine to create a pure recessive TT. This brilliantly illustrates the macro result of that microscopic sorting we just did. Because the capital T is dominant, the TT genotype and both of the TT genotypes produce a tall plant.
Only that one TT genotype produces a short plant. To put that into perspective, this gives us the classic foundational Mendelian phenotypic ratio of 3 tall to 1 dwarf, or looking at the percentages, 75% to 25%. Section 5, Avoiding Common Exam Mistakes. All right, if you are sitting for your O-level biology exam anytime soon, listen up. This is exactly where students lose the most marks, so pay close attention.
You absolutely cannot confuse these two fundamental terms. Think of it like a split screen. On the micro side, your genotype is the invisible, underlying genetic code, like TT or TT. On the macro side, your phenotype is the observable, physical manifestation of that code, like the actual tallness of the plant itself. Do not, under any circumstances, swap these terms when writing out your answers.
And here is your biggest warning. The second most common error we see is failing to account for all possible gamete combinations in the Punnett square. If you don't trace every single segregation directly into the grid, your entire mathematical ratio is going to be completely wrong at the end. You have to account for every single gamete. No shortcuts. Section 6, Final Recap and Tips. Let's do a quick fire synthesis of all these mechanics right now to lock in your understanding. Here are your 4 foundational pillars to take away. 1. Heredity is simply the passing of traits from parents to offspring. 2. Mendelian inheritance specifically focuses on the inheritance of single characteristics. 3. Punnett squares are the physical grids we use to predict genetic outcomes and specific phenotypic ratios. And 4. Your genotype is your invisible genetic makeup, while your phenotype is your visible physical trait. We'll leave you with this ultimate piece of strategy for exam day. Clearly label your diagrams and explicitly show all of your working steps to secure those full marks. It's crazy to think about, but the mechanics of the Punnett square govern literally everything, from the crops we eat to the exact traits we inherit. As you review everything we've gone over today, I'll leave you with one final thought. Knowing exactly how beautifully structured and predictable these biological calculations are, what other hidden, unexpressed traits might be lying dormant in your own genetic blueprint, just waiting for the exact right combination to appear in the very next generation?